Hello, everyone. So, chapter 4 is like a marathon, and our lecture today is as well. Today, we're talking about carbohydrates for exercise. So, with both hypoglycemia and depleted muscle glycogen may precipitate fatigue, so maintaining optimal levels of blood glucose, liver glycogen, and muscle glycogen is essential in various athletic endeavors, particularly prolonged exercise tasks. Some sport authorities indicate that carbohydrate is the master fuel for athletes. In this section, we discuss the role of carbohydrate as an energy source during exercise, the effect of training to enhance carbohydrate use for energy when needed, and various methods to provide adequate carbohydrate nutrition to the athlete before, during, and after competition, and carbohydrate intake during training. So carbohydrates supply approximately 40% of the body's total energy needs during rest, with about 15 to 20% used by the muscles. During very light exercise, fat is an important energy source. But Van Loon and others found that muscle glycogen and plasma glucose oxidation rates increased with every increment in exercise intensity. When exercise becomes more intense, such as when a person is working at 65 to 85% of capacity, carbohydrate becomes the preferred energy source. This is the crossover concept discussed in Chapter 3. At maximal or supramaximal exercise levels, carbohydrate is used almost exclusively. Thus, carbohydrate may be the prime energy source for high-intensity anaerobic events lasting for less than 1 minute and high-intensity aerobic events lasting more than an hour or two. As Holzitsky and others summarized, carbohydrate use then is associated with the intensity level of the exercise. The more intense the exercise, the greater the percentage contribution of carbohydrate. Of course, the more intense the exercise, the sooner exhaustion occurs. A fairly well-conditioned person may be able to exercise for many hours at 40 to 50% of VO2 max, for 1 to 2 hours or so at 70 to 80% of VO2 max, but only for minutes at maximal or supramaximal levels of VO2 max. As noted by Hargraves, the fatigue that occurs in a very high-intensity exercise of short duration, such as a 400-meter run, may be associated with the accumulation of hydrogen ions, a byproduct of lactic acid production. In contrast, the fatigues associated with more prolonged exercise may be connected with depleted supplies of liver and muscle glycogen, both of which may be affected by dietary practice and exercise intensity and duration. Carbohydrate intake is most important for prolonged endurance events lasting more than 90 to 120 minutes or 2 hours. Data from such endurance tasks such as the Tour de France, the bicycle race across America, and the Ironman triathlon illustrate the importance of dietary carbohydrate in sustaining high energy output for prolonged periods. Most of the athletes in these events consumed high caloric diets rich in carbohydrates both before and during competition. A classic example is the ultra-distance runner from Greece, Yanis Kouros, who won the Sydney to Melbourne race in Australia, a distance of approximately 600 miles in 5 days and 5 hours, or about 114 miles of running per day. He consumed up to 13,400 calories per day, with up to 98% being derived from carbohydrates. Cyclists in major multi-day races consume more than 800 grams of carbohydrate daily. Data obtained from exercise tasks or lesser magnitudes such as marathons of 26.2 and ultra-marathoners about 50 kilometers and longer also provide evidence of the importance of carbohydrate as the prime energy fuel, as evidenced in the review by Peters. Carbohydrate is also an essential energy fuel for prolonged sports involving many intermittent bouts of high-intensity exercise such as soccer, rugby, field hockey, ice hockey, and tennis. For example, Bangsbo noted that although soccer players are engaged in low-intensity exercise for about 70% of the time, there may be 150 to 250 bouts of brief, intense actions during a game. Athletes in these sports repeatedly use muscle glycogen stored in their fast-twitch muscle fibers, which may lead to a selective depletion in these fibers. Bangsbo identified muscle glycogen as the most important substrate for energy production in sports such as soccer. Carbohydrate is the most important energy food for exercise, besides being the only food that can be used for anaerobic energy production in the lactic acid. It is also the most efficient fuel for the oxygen system. If we look at the caloric value of carbohydrate, which is 1 gram equal to 4 calories, and fat, which is 1 gram equal to 9 calories, we might think that fat is a better source of energy. Indeed, this is so if we look at calories per gram. However, more oxygen is needed to metabolize the fat, and if we look at how many calories we get from 1 liter of oxygen, we will find that carbohydrate yields about 5.05 calories, and fat gives only 4.69. Thus, carbohydrate appears to be a more efficient fuel than fat by about 7%. Houston notes an even greater benefit if we look at ATP production. You get more ATP from glucose than you do from a fatty acid. For each unit of oxygen consumed, glucose produces 2.7 ATP, whereas palmitic fatty acid produces 2.3 ATP, a 17% difference. The metabolic pathways for carbohydrate are also more efficient than those for fat. In essence, during aerobic glycolysis, carbohydrate is able to produce ATP for muscle contraction up to 3 times more rapidly than fat, and even faster during anaerobic glycolysis. So the primary carbohydrate source of energy for physical performance is muscle glycogen, specifically the glycogen in the muscles that are active. Elite marathon runners may use about 4 to 5 grams of carbohydrate per minute. As the muscle glycogen is being used during exercise, blood glucose enters the muscles and the energy pathways. In turn, the liver will release some of its glucose to help maintain or elevate blood glucose levels and prevent hypoglycemia. Coyle noted that during moderate exercise, muscle glycogen and liver glycogen contribute equally to carbohydrate oxidation. At higher intensities, muscle glycogen use increases. Thus, all body stores of carbohydrate, blood glucose, liver glycogen, and muscle glycogen are important for energy production during various forms of exercise. Proper physical training is essential to optimize carbohydrate utilization during exercise, as is proper carbohydrate nutrition. Because carbohydrate is a primary fuel for exercise, as you initiate an endurance exercise program, a major proportion of your energy will be derived from your muscle glycogen stores. A single bout of exercise can activate the genes that produce GLUT4 receptors, which can exert an insulin-type effect by facilitating the transport of blood glucose into the muscle both during and immediately following the exercise bout. As you continue your endurance exercise program, such as running or bicycling, the physical activity serves as a metabolic stressor to the body, and various tissues begin to adapt to better accommodate the exercise stress. Houston reported that as few as 5 days of training can exert favorable effects, such as decreasing the production of lactic acid for a standardized exercise task. These adaptations have implications for physical performance and the fuels used. So the following have been noted to occur in both males and females, young and old, after several months of endurance training. Of equal or greater importance, you will be able to work at a greater percentage of your VO2 without fatigue. Endurance-trained muscles may use less glucose at a low-intensity exercise, but have the capacity to use more during intense or maximal exercise. Endurance-trained muscle has an increased maximal capacity to utilize carbohydrates. As muscle cell mitochondria density increases, the enzymes that metabolize carbohydrate in the muscle cells will increase, especially oxidative enzymes associated with the Krebs cycle. As we shall see in Chapter 5, training also enhances the use of fat during exercise. By doing so, there is a less reliance on carbohydrate oxidation during submaximal exercise. More glycogen is stored in the muscle as another benefit. Synthesis of muscle glycogen may be twofold, faster in trained versus untrained individuals. So what do all these changes mean? You may be able to run a 10-kilometer or 6.2 miles road race at a 7-minute per mile pace instead of 8 minutes. You can cruise in high gear for longer periods because you have increased your ability to produce energy from carbohydrates. Also, by reducing your reliance on carbohydrates at lower running speeds, you may compete in more prolonged races such as marathons without becoming hypoglycemic. Some of the effects of aerobic or endurance training upon skeletal. These are some of the effects of aerobic or endurance training upon skeletal muscle. You'll see an increase in glycogen and triglycerides provide a greater energy store. Increased levels of GLUT4 receptors provide the potential to increase glucose delivery to the muscle as needed, while the increase in mitochondrial size and number, as well as myoglobin content, oxidative enzymes, and slow twitch muscle fiber size, facilitates the use of oxygen for production of energy. So how is hypoglycemia related to the development of fatigue? As noted previously, blood glucose is in very short supply, so it is being used during exercise. It must be replenished from liver glycogen stores. A depletion of liver glycogen may lead to hypoglycemia during high-intensity aerobic exercise because gluconeogenesis normally cannot keep pace with glucose utilization by the muscles. Hypoglycemia is known to impair the function of the central nervous system and is often accompanied by acute feelings of dizziness, muscular weakness, and fatigue. The normal blood glucose level usually ranges from 80 to 100 milligrams of glucose per 100 milliliter of blood. As this level gets progressively lower, hypoglycemic symptoms may develop. The point usually used to identify hypoglycemia during research studies with exercise is 45 milligrams per 100 milliliters or 2.5 millimoles per liter, although some investigators have used higher. Because hypoglycemia disrupts functioning of the central nervous system, the brain, and the spinal cord, the body attempts to maintain an optimal blood glucose level. Zierler noted that exercise increases muscle glucose uptake, in part due to increased cell membrane GLUT4 receptors. Exercise may also increase the sensitivity to insulin, so more glucose is transported into the muscle for the same level of insulin. Thus, insulin levels normally drop during exercise so as to help maintain normal serum glucose. Other hormones, epinephrine, adrenaline, glucagon, and cortisol also help maintain and even increase blood glucose levels during exercise. Epinephrine is secreted from the adrenal gland during exercise, particularly intense exercise, and stimulates the liver to release glucose. It also accelerates the use of glycogen in the muscle. However, during the early phases of exercise, liver glucose output may exceed muscle glucose uptake, which may result in hyperglycemia. Glucagon and cortisol levels generally increase during the stress of exercise, cortisol particularly during prolonged exercise. Glucagon is released from the pancreas and generally increases the rate of gluconeogenesis in the liver. Kyer noted that liver gluconeogenesis may contribute substantially during prolonged aerobic exercise when liver glycogen levels decline and gluconeogenesis substrate becomes more abundant. Cortisol is a hormone that's secreted from the adrenal gland and facilitates the breakdown and release of amino acids from muscle tissue to provide some substrate to the liver for gluconeogenesis. Blood glucose normally increases during the initial stages of exercise and is normally well maintained by these hormonal mechanisms. And there's a summary of hormonal actions in the regulation of blood glucose that's presented in table 4.6. So here, again, you can see epinephrine, glucagon, and cortisol, and where the gland that secretes it, as well as the stimulus and the action that it provides. So how is lactic acid production related to fatigue? In review, as noted in Chapter 3, lactic acid is an end product of anaerobic glycolysis. Anaerobic glycolysis represents the lactic acid energy system found primarily in the type 2B white muscle fibers, but also the type 2A red fibers. Anaerobic glycolysis is increased at the onset of high-intensity exercise, such as 200 to 400 meter track events, as a means to rapidly replenish ATP for muscle contraction or for the rapid resynthesis of PCR. The process of aerobic glycolysis can also generate ATP, but it is too slow during high-intensity exercise. Thus, lactic acid or lactate is produced when the production of pyruvate from glycolysis exceeds the oxidative capacity of the mitochondria. Blood lactic acid or lactate levels increase during high-intensity exercise and for years have been thought to be the cause of fatigue. However, Coyle notes that the lactate molecule per se does not cause fatigue, but rather that its accumulation in blood reflects a disturbance of muscle cell homeostasis. One factor that may disturb muscle cell homeostasis is an increased concentration of hydrogen ions, reflecting an increased acidity. DeBold notes that even after 100 years of research on muscle fatigue, the molecular basis is still poorly misunderstood. Using in vitro studies, DeBold has demonstrated that high levels of hydrogen ions and phosphate inhibit the molecular motions of muscle proteins such as myosin, troponin, and tropomyosin, which leads to a decreased force production and potentially fatigue. Fitz noted that it is now generally thought that the component of fatigue correlated with lactate results from the effects of an increased free hydrogen ion rather than lactate or the undissociated lactic acid. The hydrogen ion and associated increased acidity could elicit fatigue by inhibiting a number of physiological processes in the muscle cell associated with contraction. As noted in Chapter 13, sodium bicarbonate, a buffer of acidity in the muscle cell, may be an effective means to enhance performance in high-intensity exercise tasks. The lactate produced during exercise is not a waste product. Lactate is a carbohydrate. One molecule of lactate contains about half the energy of one molecule of glucose. Years ago, George Brooks from the University of California at Berkeley proposed the lactate shuttle, whereby the lactate produced during exercise in white muscle fibers would be shuttled to other tissues, such as the heart and red oxidative muscle fibers, where it could be oxidized for energy. Hashimoto and Brooks also hypothesized the lactate produced in the cell cytoplasm may be oxidized by mitochondria in the same cell. Research supports the lactate shuttle hypothesis, and as discussed later in this chapter, some sports scientists have actually tested lactate salt supplementation as a means to provide energy to enhance exercise performance. So how is low muscle glycogen related to the development of fatigue? Low muscle glycogen may impair both aerobic and anaerobic exercise performance. Low muscle glycogen in aerobic exercise is where muscle glycogen is the major energy source for prolonged, moderately high intensity aerobic exercise. Elite marathon runners can maintain a fast race pace primarily through oxidation of carbohydrate, primarily muscle glycogen, and to a lesser extent fatty acids stored in the muscle. Coyle indicates that exercise at 70 to 85% of VO2 max cannot be maintained without sufficient carbohydrate oxidation, and thus the severe lowering of muscle glycogen, often coupled with hypoglycemia, resulting in the need to reduce exercise intensity to 40 to 60% of VO2 max. A number of studies have shown that physical exhaustion was correlated with very low muscle glycogen levels, but others have shown glycogen remaining even though subjects were exhausted. Several mechanisms have been postulated to explain the development of fatigue even with some muscle glycogen remaining. For one mechanism would be the location of glycogen. Costill has indicated that performance would be adversely affected only when muscle glycogen levels went below 40 millimoles of kilogram of muscle tissue. It may be that complete depletion of muscle glycogen is not necessary for performance to suffer, for glycolysis may be impaired with low glycogen levels, or the glycogen in the muscle fiber may be located where it is not readily available for glycolysis. Ortenblad and colleagues have described that glycogen is not homogeneously distributed in muscle fibers. It is localized in separate pools. Additionally, glycogen granules have their own glycolytic enzymes and regulating proteins, and maybe the depletion of these complexes localized in the myofibrils that is related to fatigue. Another mechanism would be rate of energy production. Schulman and Rothman propose a model in which energy is supplied in milliseconds via glycogenolysis, and indicate that one possible mechanism for muscle fatigue is that at low but non-zero glycogen concentrations, there is not enough glycogen to supply millisecond energy needs. In a related vein, Fitz noted that low levels of muscle glycogen may interfere with maintenance of optimal levels of Krebs cycle intermediates, which can reduce the rate of aerobic ATP production, and further notes that the metabolism of blood-borne substrates, blood glucose and free fatty acids, is simply too low to maintain heavy exercise intensities. Other mechanisms would be muscle fiber type. The fatigue that develops may be related to the depletion of muscle glycogen from specific muscle fiber types. And prolonged exercise at 60 to 70% of VO2 max, type 1 fibers, which are red, oxidative, slow twitch, and type 2A fibers, red, oxidative, glycolytic, fast twitch, are recruited during the early stages of the task. But as muscle glycogen is depleted, the athlete must recruit type 2B fibers, which are white, glycolytic, fast twitch, to maintain the same pace. However, it takes more mental effort to recruit the type 2B fibers, which will be more stressful to the athlete. Type 2B fibers also are more likely to produce lactic acid, increasing the acidity level, which may increase the perceived stress of the exercise. In a study, Krustup and others reported that glycogen depletion of the slow twitch muscle fibers necessitating recruitment of fast twitch muscle fibers and increased energy demands is a factor that may predispose to fatigue. Another mechanism would be use of fat for energy. As muscle glycogen becomes depleted in the slow twitch muscle fibers, the muscle cell will rely more on fat as the primary energy source. Because fat is a less efficient fuel than carbohydrate, the pace will slow down. Another mechanism is the role of the brain. Signals sent from peripheral tissues to the brain may regulate energy metabolism. A low glycogen level in exercising muscles may be such a signal and may invoke neural responses causing fatigue. Matsui and colleagues recently showed decreased liver and muscle glycogen after 30 and 60 minutes of running, and a decrease in glycogen in 5 areas of the brain after 120 minutes of running, or 2 hours. The interactions between decreased muscle and brain glycogen and fatigue need to be studied further. Low muscle glycogen and anaerobic exercise. Fatigue is very high intensity anaerobic type exercise generally is attributed to the detrimental effects of the acidity in the muscle cell associated with lactic acid production. Research has shown that maximally high intensity exercise lasting only about 60 seconds is not impaired by a very low muscle glycogen concentration, approximately about 30 millimoles per kilogram of muscle. However, it is possible that performance in such very high intensity short-term exercise tasks may be impaired with extremely low muscle glycogen in the fast twitch muscle fibers. Moreover, with somewhat longer anaerobic tasks, approximately 3 minutes, one laboratory study reported a reduced performance in the time to exhaustion test after 4 days of a low carbohydrate, high fat diet when compared to a normal mixed diet and a high carbohydrate diet. Although muscle glycogen levels were not measured, a logical assumption is that they were lower on the low carbohydrate diet. In addition, field research has suggested that slower overall sprint speeds, such as the latter parts of prolonged athletic contests, like soccer and ice hockey, may be due to muscle glycogen depletion. Muscle biopsies of these athletes revealed very low glycogen levels, which were attributed not only to the strenuous exercise in the contest, but also to the fact that these athletes were consuming diets low in carbohydrates. In support of these field studies, Balsam and others reported that low muscle glycogen levels impaired laboratory exercise performance in repeated bouts of very high intensity intermittent exercise. 6-second cycle ergometer performance followed by 30 seconds of rest. Krustup and others reported that almost 50% of muscle fibers were completely or almost empty of glycogen following a soccer game, and suggested that a slower sprint performance in the latter part of a game may be explained by low glycogen levels in individual muscle fibers. Also, low muscle glycogen stores may lead to a decrease in exercise intensity during training. In summary, low levels of glycogen in the white, fast twitch 2B muscle fibers may limit performance in the intermittent anaerobic type aerobics exercise task. Both hypoglycemia and low glycogen in the red muscle fiber types, most likely a combination of the two, may be a contributing factor to fatigue and prolonged endurance exercise. So how are low endogenous carbohydrate levels related to central fatigue hypothesis? As noted previously, hypoglycemia and low muscle glycogen levels may impair exercise performance, and one mechanism for each involves adverse effects on brain function. Collectively, they may contribute to central fatigue in a different way. In the latter stages of prolonged exercise bouts, low muscle glycogen in combination with the decreased blood glucose levels will stimulate gluconeogenesis from muscle protein. In particular, branched chain amino acids, or BCAAs, in the muscle will be catabolized to provide energy. Because BCAA release from the liver may be decreased or uptake by the muscle may increase, blood levels of BCAA decline. The central fatigue hypothesis during prolonged exercise suggests that this decline in blood BCAA may contribute to fatigue. In general, fatigue is hypothesized to occur when BCAA levels drop and the concentration of another amino acid, tryptophan, increases in its free form, or free tryptophan. BCAAs complete with free tryptophan for similar receptors compete with free tryptophan for similar receptors that facilitate their entry into the brain. So, high BCAA levels prevent brain uptake of free tryptophan. With an increased free tryptophan BCAA ratio, entry of free tryptophan into the brain cells will be facilitated. Increased brain levels of tryptophan may stimulate the formation of serotonin, a neurotransmitter in the brain that may be related to fatigue sensations. Preventing the increase in the free tryptophan BCAA ratio is theorized to prevent the premature development of fatigue, and the use of BCAA supplements in this regard will be covered in Chapter 6. Musin has noted that postponing central fatigue with nutritional interventions, including amino acids, water, carbohydrate, and caffeine, has been studied. Although carbohydrate and caffeine show promise in reducing central fatigue, other nutrients do not appear as promising. However, carbohydrate intake during exercise may also be helpful. So this is a diagram showing the central fatigue hypothesis and looking at BCAA and the central fatigue hypothesis. And in A, you see an increased level of serum free tryptophan, resulting in a high free tryptophan BCAA ratio that will bind to receptors in the capillary for transport to nerve cells, and increased serotonin formation, which may be associated with central fatigue. If you look at B, there's an increased level of BCAAs that will compete with the free tryptophan for receptors, blocking them from entering the nerve cell and decreasing production of serotonin, helping to prevent central fatigue. So, will eating carbohydrates immediately before or during an event improve physical performance? Because hypoglycemia or muscle glycogen depletion may be a cause of fatigue during endurance exercise, supplementation with glucose or other forms of carbohydrate before or during exercise may be theorized to delay the onset of fatigue and improve performance. Thousands of studies have been conducted on this topic ever since carbohydrates were identified as the most efficient energy source for exercise more than 80 years ago. And researchers' interest in this topic remains unabated today. In recent years, the research designs have usually been highly sophisticated as investigators have attempted to provide specific answers relative to the type, amount, and timing of carbohydrate ingestion before and during performance. Although some problems remain in providing quantitative data, the use of stable isotopes of ingested carbohydrates, referred to as exogenous carbohydrates, in comparison to endogenous stores in the body, as detailed by Wolf and George, has enhanced our understanding of their metabolic fate when ingested prior to or during exercise. However, the reviewer attempting to synthesize the available research is confronted with a difficult task, as the experimental designs varied considerably. The amount and type of carbohydrate ingested, the use of liquid form or solid forms, the method of administration, whether it's oral ingestion or venous infusion, the time prior to or during the exercise that it was taken, the diet of the subject several days prior to the study, the amount of glycogen in the muscle and liver, the intensity and duration of this exercise, the type of exercise task, whether it's running, swimming, or cycling, the fitness level of the subjects, the environmental temperature, and the method used to evaluate blood glucose and muscle glycogen utilization are some of the important differences between the studies. Although the results from all of these studies were not similar, some general consistencies have evolved. The role of carbohydrate supplementation on exercise performance has been the subject of numerous reviews, and a number of contemporary reviews may be found in the reference list at the end of the chapter. Based on these reviews and an overall review of specific studies, following generalizations appear to be logical. More specific information relative to practical recommendations is provided following this discussion. So the use of ingested carbohydrate. So using labeled carbohydrate sources and analyzing expired carbon dioxide for radioactivity. Investigators have shown that some of the ingested or exogenous carbohydrate may be used as an energy source within 5 to 10 minutes, indicating that it may empty rapidly from the stomach, be absorbed from the small intestine, and enter the metabolic pathways. Peak use of exogenous carbohydrate appeared to occur 75 to 90 minutes after ingestion. A number of studies have shown that the ingested carbohydrate may contribute a significant percentage of the carbohydrate energy source during exercise, ranging from 20 to 40% in some studies, but as much as 60 to 70% during the later stages of exercise, when endogenous liver and muscle glycogen stores become depleted. So will eating carbohydrates immediately before or during an event improve physical performance? Because hypoglycemia and muscle glycogen depletion may be a cause of fatigue during endurance exercise, supplementation with glucose or other forms of carbohydrate before or during exercise may be theorized to delay the onset of fatigue and improve performance. And that seems to be a repeat. So, possible fatigue delaying mechanisms. The precise mechanism whereby glucose ingestion helps delay the onset of fatigue during moderate to high intensity exercise, which is greater than 65% VO2 max, has not been totally elucidated, but several theories have been studied. Maintenance of blood glucose levels is another possible fatigue delaying mechanism. The available data suggests that the ability of carbohydrate intake to delay fatigue may be related to the maintenance of higher blood glucose levels, possibly by sparing liver glycogen until late in the exercise and the prevention of hypoglycemia in susceptible individuals. Blood glucose would be available to enter the muscle and provide a source of energy for aerobic glycolysis, and may provide glucose to the brain to prevent premature central fatigue. As noted, exogenous glucose is used increasingly as the exercise task becomes prolonged. Another possible fatigue delaying mechanism is the reduction of psychological effort. Some research has shown that glucose ingestion could make an endurance task psychologically easier, and suggested that the physiological effects of the glucose, either in the brain or in the muscles, reduces the stressful effects of exercise. Several studies by Utter and others found that carbohydrate ingestion reduced the ratings of perceived exertion during prolonged running and cycling. For example, they reported that marathoners ingesting carbohydrate compared to placebo beverages were able to run at a higher intensity during a competitive marathon, and yet the ratings of perceived exertion were similar in both groups of runners, suggesting that the carbohydrate may have permitted them to run at a faster rate with similar psychological effort. However, it has not been determined whether these ergogenic effects may be attributed to the effect of glucose as a source of energy in the muscle, or to its effect on the central nervous system, either as a direct energy source for brain metabolism or through its effect on BCAA levels. In an effort to improve endurance performance, Druckendrup pioneered a method of rinsing but not ingesting carbohydrate in the mouth. It has been shown that the presence of carbohydrate in the mouth activates several areas of the brain. In a recent review, he highlighted 12 studies that examined the effects of carbohydrate mouth rinse on endurance exercise performance, looking at 30 to 70 minutes, and reported that 9 studies showed significant improvements. These improvements were of a similar magnitude as would be expected with carbohydrate ingestion. The carbohydrate mouth rinse methods might be particularly useful for athletes who experience gastrointestinal upset with carbohydrate ingestion, or those attempting to reduce energy intake to manage their body mass. In several studies, carbohydrate supplementation during exercise has been reported to prevent the decrease in serum BCAA during the later stages of prolonged exercise, possibly by mitigating the secretion of cortisol, according to the central fatigue hypothesis, preventing an increase in the free tryptophan BCAA ratio that would deter the onset of mental fatigue. Another mechanism for possibly delaying fatigue would be sparing of muscle glycogen. Although sparing of muscle glycogen could be another benefit of carbohydrate ingestion before or during moderate to high intensity exercise, research findings are equivocal. In a unique experiment from the University of Texas, subjects received venous glucose infusions to maintain a hyperglycemic state during 2 hours of exercise at 73% of VO2 max, but the net rate of muscle glycogen utilization was not affected compared to control conditions. Arkansas and Chrysanthopoulos and their colleagues also noted no muscle glycogen sparing effect with carbohydrate supplementation during the exercise task. In contrast, Yaspekulis and others, also from the University of Texas, noted that during low-intensity exercise, less than 50% VO2 max, or during low to moderate intensity exercise tasks, carbohydrate supplementation during exercise could spare use of muscle glycogen in slow-twitch muscle fibers and enhance performance. They noted that during low-intensity exercise, the serum levels of both glucose and insulin were elevated, which could promote muscle use of serum glucose and sparing of muscle glycogen. Bosch and others, as well as Tensas and others, reported that carbohydrate intake during prolonged exercise at about 70% max spare muscle glycogen use, and the Tensas group reported that the sparing effect occurred in the type 1 slow-twitch muscle fibers, but not in the type 2 fast-twitch muscle fibers. Williams noted that although other reviewers concluded that consuming carbohydrate during exercise did not spare use of muscle glycogen in cyclists, research findings from his laboratory did find glycogen sparing in runners who consumed carbohydrate drinks throughout prolonged exercise, and the glycogen sparing occurred early in the exercise task. Hargraves noted also that the breakdown of muscle glycogen may be slowed because the supply of blood glucose is improved when carbohydrate is consumed. There are limitations to prevent fatigue. Although glucose ingestion may help delay fatigue during moderately high intensity exercise, it cannot totally prevent the onset of fatigue. It appears that a maximum of about 1.5 to 1.7 grams of the ingested carbohydrate may be available each minute, which is much lower than the required energy needs at 65 to 85% of VO2 max. Jukendrup and Jensen indicate that the intestines and liver may be limiting factors. The intestines being unable to absorb the ingested carbohydrate at a faster rate, while the liver may limit the amount of glucose released into the blood when blood glucose, BCAA, and or muscle glycogen levels are eventually reduced to a critical level. Fatigue occurs. Will eating carbohydrate immediately before or during an event improve physical performance? If the individual has a normal liver and muscle glycogen stores, glucose feedings are unnecessary for continuous moderately high intensity exercise bouts lasting 60 to 90 minutes or less, but may be beneficial for high intensity exercise tasks of similar duration, as noted below. Because the body can store carbohydrate in the muscles and liver, the usefulness of glucose or other carbohydrate intake before or during exercise depends on the adequacy of those supplies already in the muscle and liver to meet energy needs. For competition, the muscle and liver glycogen stores should be adequate to meet carbohydrate energy needs. The critical point to consume substantial amounts of carbohydrates a day or two prior to the event and to decrease the duration and intensity of training to assure ample endogenous glycogen supplies. So will eating carbohydrate immediately before or during an event? We're continuing with that. The available research has shown that the consumption of glucose, fructose, sucrose, maltodextrin, a glucose polymer, or other carbohydrate combinations immediately prior to events of short or moderate duration has a negligible effect upon performance. Adding a gallon of gas to a full tank will not make a car go faster during a short ride. The same is true of sugar to a muscle already filled with glycogen. If, however, muscle glycogen levels are low and the exercise task is somewhat prolonged, then ingestion of carbohydrate just prior to the exercise bout may improve performance. It is important to note, however, that to enhance performance, the exogenous carbohydrate source must be able to delay the onset of fatigue that might otherwise occur as a result of premature depletion of endogenous carbohydrate sources. Exercise intensity and duration If we look at the effects of exogenous carbohydrate on exercise performance of a different intensity and duration, the potential beneficial effects of carbohydrate supplementation depend on the interaction of exercise intensity and duration, which of course are interrelated. The shorter the duration, the greater the exercise intensity can be. Stillingworth and Cox have written the most comprehensive review of the effects of carbohydrate ingestion on exercise performance that takes into account the influence of exercise intensity and duration. They reported that of 61 total studies, 50 or 82% showed a performance-enhancing effect of carbohydrate ingestion. Of 10 studies that assessed the effects of carbohydrate ingestion on exercise tasks lasting less than an hour, 6 showed a performance enhancement. Of 13 studies of the effects of carbohydrate mouth rinse on exercise performance lasting less than 1 hour, 10 showed a beneficial effect. During exercise tasks lasting from 60 to 120 minutes, 15 of 18 studies showed a performance-enhancing effect of carbohydrate ingestion. Finally, in studies with exercise tests lasting more than 120 minutes in duration, 16 of 17 showed a performance-enhancing effect of carbohydrate supplementation. Overall, there was a positive relationship between total exercise time and percent increase in performance, indicating that carbohydrate ingestion was more effective in longer tasks. The authors noted that the primary mechanisms through which carbohydrate improves performance probably differed based on exercise intensity and duration. For instance, during brief, intense exercise, carbohydrate acts on the receptors in the mouth and works through the central nervous system to enhance performance. Thus, the type of carbohydrate may not be important. And longer exercise tasks rapidly absorbed carbohydrates, possibly greater than 90 grams per hour, are needed to replenish depleting muscle glycogen stores. The authors stressed that recommendations should be tailored to individual tolerance. The following sections provide more details about some of the individual studies that have evaluated the effects of carbohydrate ingestion on exercise performance. So the effects are dependent upon very high intensity exercise for less than 30 minutes. The research suggests that carbohydrate supplementation will not enhance performance in high intensity exercise bouts less than 30 minutes in length. For example, Palmer and others noted that consuming carbohydrate 10 minutes before competing in a 20-kilometer cycle time trial did not enhance performance in well-trained cyclists. Nevertheless, if carbohydrate supplements could ameliorate a muscle or liver glycogen deficiency, performance could improve. For example, Wahlberg, Rankin reported that carbohydrate supplementation improved high-intensity anaerobic exercise performance in wrestlers following a drastic weight reduction program with very limited carbohydrate intake. Presumably, the carbohydrate supplement consumed in a 5-hour period before testing increased muscle glycogen levels, particularly in fast twitch fibers, enhancing carbohydrate utilization and subsequent performance. But with very high intensity resistance exercise training, while the performance enhancing benefits of carbohydrate ingestion on endurance exercise performance are well accepted, less is known about the effects of carbohydrate on resistance training performance. As strength and conditioning training professionals are an integral part of success in most sports, properly fueling resistance training workouts is essential. A bout of resistance exercise can reduce muscle glycogen by up to 40%, indicating that the importance of glycogen as a fuel for resistance training and raising the possibility that carbohydrate ingestion or mouth rinsing may improve resistance exercise performance. With high-intensity exercise for 30 to 90 minutes within this time frame, the potential benefit of carbohydrate consumption may depend on the duration of the exercise, intensity of exercise, and training level of the athlete. Moreover, supplementation may benefit well-trained athletes who may be able to exercise at high intensity for about an hour. As indicated earlier, the presence of carbohydrate in the oral cavity has been shown to improve the performance of stimulating the central nervous system. Exercises lasting about an hour have been the most well-studied, and it appears the type of sweetness of carbohydrate is not relevant. Currently, the most practical advice points towards swishing 1.5 grams of carbohydrate per milliliter of water for 5 to 10 seconds during every 8 to 10 minutes of exercise. At this time, a great deal more work needs to be done to learn how carbohydrate mouth rinsing improves exercise performance and what conditions may enhance or reduce these effects. So when and how much and what form should carbohydrates be consumed before or during exercise? The most common athletic events or physical performance activities that may benefit from carbohydrate feedings are those associated with long duration, 90 to 120 minutes or more, at a moderate to high intensity level. Marathon running, cross-country skiing, and endurance cycling are common sports of this kind. Other sports that require intermittent bouts of intensive activity over a prolonged period, such as soccer, may also benefit. However, the individual participating in these events, particularly under warm or hot environmental conditions, also needs to replenish fluid losses incurred through sweating. In such cases, fluid replenishment is more critical than carbohydrate. The topic of fluid replacement during exercise is covered in more detail in Chapter 9, but because carbohydrate is one of the contents in the majority of sports drinks developed as fluid replacements for athletes, its role is discussed briefly here. Again, based on current reviews by the primary investigators regarding carbohydrate supplementation for exercise performance and a careful analysis of individual studies, the following points represent the general conclusions and recommendations for individuals who may be exercising at 60 to 80% of their VO2 max or greater for 1 to 2 hours or longer. These points may also be applicable to athletes engaged in intermittent, high-intensity exercise sports that last an hour or more. But remember, individuals may have varied reactions to carbohydrate intake. So, athletes should experiment in training before using these recommendations in actual competition. So, if before exercise, when and how much? Looking at 4 hours or less before exercise, carbohydrate intake may enhance performance. Research has demonstrated improved performance when adequate carbohydrate was consumed either 1, 3, or 4 hours prior to a prolonged exercise task, involving simulated racing conditions during the latter stage. Other research revealed no significant differences in 30-kilometer run performance when equal amounts of carbohydrate were supplemented, either 4 hours before or during the run, suggesting the ingested carbohydrate rate was available for energy production use either strategy. The amount of carbohydrate ingested 4 hours prior to performance should be based upon body weight. Several studies have used 4 to 5 grams per kilogram or 1.8 to 2.3 grams per pound with good results. For an athlete who weighs 60 kilograms or 132 pounds, the recommended amount would be 240 to 300 grams. The carbohydrates should be consumed in any of several forms, including fluids such as juices, glucose polymer solutions, or solid carbohydrates such as fruits or starches. The fiber content should be minimized to prevent possible intestinal problems during exercise. Keep in mind that 300 grams of carbohydrate is about 1,200 calories, a somewhat substantial meal. You may consult Appendix E for an expanded list of foods in carbohydrate. So, if you look at less than 1 hour before exercise, Jukendrup and Killer reviewed the myths surrounding pre-exercise carbohydrate feedings. They found that the ingestion of carbohydrates in the hour before exercise either improved performance or had no impact on performance. Based on these findings, there is little evidence to abstain from carbohydrate ingestion in the hour prior to exercise for those who do not experience some symptoms of hypoglycemia. Prudence suggests that individuals who may be prone to reactive hypoglycemia should avoid carbohydrate intake, particularly high glycemic index foods, 15 to 60 minutes prior to performance. Simple sugars ingested with this time frame may actually impair physical performance in such individuals because of the adverse effects of reactive hypoglycemia, such as muscular weakness. Moreover, this same insulin response may speed up muscle glycogen utilization. This may be a disadvantage to the marathoner, whose glycogen levels may be depleted too early in the race. Several earlier studies showed that run time to exhaustion was shorter by about 20 to 25% after athletes consumed 2 to 3 ounces of glucose within an hour before the endurance test. However, not all individuals experience reactive hypoglycemia. If carbohydrate is consumed approximately 1 hour prior to performance, about 1 to 2 grams per kilogram or 60 to 120 grams for a 60 kilogram athlete may be recommended. 4 of these levels have been shown to enhance performance in several studies. One study using only 12 grams 1 hour prior to performance showed no beneficial effect. Both glucose polymers and foods with a high glycemic index have been used successfully. What about immediately before exercise? As noted previously, consuming carbohydrate immediately before exercise of short duration, and even exercise tasks of less than 90 minutes or so normally will not enhance performance. For example, Marison and others found that the ingestion of carbohydrate solution 30 minutes before undertaking 30-second anaerobic wind gait tests had no effect on power output. Smith and others reported no significant effect on swim time performance of ingesting a 10% glucose solution 5 minutes before a 4-kilometer swim of approximately 70 minutes. However, carbohydrate intake immediately prior to within 5 to 10 minutes prolonged endurance exercise tasks of 2 hours or more may help delay the development of fatigue and may improve performance if the athlete is exercising at a level greater than 50% VO2 max, such as 60 to 75%. The majority of the studies included controlled laboratory investigations and field research involving different types of endurance athletes support this point of view. At this level of exercise intensity, the insulin response to glucose ingestion is suppressed. In addition, the secretion of epinephrine is increased. These two hormonal responses, in addition to the secretion of epinephrine, is increased. These two hormonal responses interact to help maintain or elevate the blood glucose level and prevent the hypoglycemic response that typically may occur in reactive individuals if more time elapses between the ingestion of the carbohydrate and the initiation of exercise. If carbohydrates are consumed immediately before exercise, that is within 10 minutes of the start, about 50 to 60 grams of glucose polymer in a 40 to 50% solution have been used effectively in some studies. Dry glucose polymers are available commercially. 1 tablespoon is about 15 grams. To make a 50% solution containing 50 grams of polymer, put about 3 level tablespoons of the polymer into 100 milliliters, about 3 to 4 ounces of water. To make a 7.5% solution containing 15 grams, put 1 tablespoon of the polymer into 200 milliliters or 7 ounces of water. Several commercial energy drinks contain 25 to 50 grams of carbohydrate per 8 gram fluid ounces, which are about 10 to 20% solutions. So 4 hours before you have a 70 kilogram runner, they should ideally take in about 280 to 350 grams. And they would look something very much like this: a large bagel, turkey breast, orange juice, yogurt, banana, an energy drink, something along that line. 4 hours prior, so it would be like 4 to 5 grams per kilogram. You should have some lean protein. If it's an hour before, 1 to 2 grams per kilogram, and immediately after, you want to take in about 1 gram per kilogram. And this is just looking at the recommended guidelines for carbohydrate intake as a means to help optimize exercise sports performance. Carbohydrate intake before and during exercise may help optimize performance, whereas carbohydrate intake after exercise may facilitate recovery for subsequent training or competition. In general, the lower the intensity and the shorter the duration of exercise, the less need for additional carbohydrate. Recommendations are based on scientific studies, as we've been discussing. Physically active individuals should consume various types and amounts of carbohydrate before and during exercise and sports training to determine personal optimal dietary strategies. And this just gives you a look at a quick estimate of carbohydrates in the various food exchanges and sports products. So if we look at carbohydrate intake during exercise, carbohydrate ingested during prolonged exercise can help maintain blood glucose levels and reduce the psychological perception of effort, as measured by the ratings of perceived exertion during the latter stages of an endurance task. As the exercise task continues and the muscle glycogen level falls, the amount of energy derived from the ingested carbohydrate increase. Most research supports the benefits of consuming carbohydrate early in and throughout the exercise task. But even a single carbohydrate feeding late in a prolonged exercise bout may help replenish blood glucose levels, increase carbohydrate oxidation, and delay fatigue. So if you supplement during exercise, when and how much? During exercise, feedings every 15 to 20 minutes appear to be a reasonable schedule, but possibly more frequently when attempting to maximize carbohydrate intake or to obtain fluids when exercising under warm or hot environmental conditions. Although you may consume considerable quantities of carbohydrate during exercise, your ability to use this exogenous source for energy is limited. The reason is not known, but as noted previously, may be related to insufficient intestinal absorption or impaired delivery from the liver. Sports drinks averaging 6 to 10% carbohydrate have been found to enhance prolonged endurance performance. A typical serving of a sports drink, about 8 ounces, would contain about 14 to 24 grams of carbohydrate. So, depending on the concentration, an athlete who wanted to maximize utilizable carbohydrate intake would need to drink about 32 to 56 ounces to obtain 100 grams of carbohydrate per hour. Drinking 8 ounces every 15 minutes would provide 32 ounces or 1 quart over the course of an hour, but consumption would have to be more frequent to obtain 56 ounces. Consuming 56 ounces of fluid over the course of an hour might be difficult and may pose a potential health risk for some individuals. Although the fluids could provide the desired amount of carbohydrate, excessive fluid consumption could lead to overhydration and a serious medical condition known as hyponatremia, which is discussed in Chapter 9. So optimal supplementation, if we look at several studies have indicated that although the intake of carbohydrate either before or during exercise may separately enhance performance, the best effect was observed when carbohydrate was consumed both before and during exercise. For example, Chrysanthopoulos and others had subjects run to exhaustion at 70% VO2 max and found that although a high carbohydrate meal 3 hours prior to performance improved endurance time, the combination of the meal and a carbohydrate electrolyte solution during exercise further improved endurance running capacity. Asker Jukendrup, Naomi Cermak, and Luc Van Loon recommend the following guidelines for carbohydrate intakes during exercise to maximize performance and minimize gastrointestinal distress. For athletes engaged in very short, high-intensity exercise lasting less than half an hour, no carbohydrate is necessary during exercise. For short and high-intensity exercise or intermittent team sports lasting half an hour to 1.25 hours, very little carbohydrate is required, possibly just a mouth rinse. For intermittent team sports of moderate duration from 1 to 1.5 hours, up to 60 grams per hour is recommended. For intermittent team sports of long duration beyond 2 hours, up to 90 grams per hour of multiple transportable carbohydrates or mixed sugars are recommended. For endurance, continuous exercise lasting 1 to 3 hours, up to 60 grams per hour of carbohydrate are recommended. And finally, for prolonged endurance exercise beyond 2.5 hours in duration, up to 90 grams per hour of multiple transportable carbohydrates are recommended. Additionally, it is recommended that if an athlete is going to exercise for more than 2 hours, but is starting exercise with suboptimal carbohydrate levels, they consume up to 90 grams of multiple transportable carbohydrates per hour. Athletes may learn to tolerate higher amounts of carbohydrates, so these recommendations, although based in research, allow for some adjustment. Athletes should experiment with different doses of carbohydrate during training or practice, but not competition. So if we look at the different types of carbohydrate combinations, a number of different types of carbohydrates have been studied, including glucose, fructose, galactose, sucrose, maltose, glucose polymers such as maltodextrins, both individually and in various combinations, as well as soluble starch, a very long polymer, high glycemic index foods such as potatoes, and low glycemic index foods such as legumes in general. There appears to be no difference between these different types of carbohydrates as a means to enhance endurance performance when used properly. However, there may be important considerations relative to the use of various carbohydrate combinations, fructose, solid carbohydrates, and low glycemic index foods. with carbohydrate combinations, Druckendrup noted studies showing that a single carbohydrate ingested during exercise will be oxidized at rates up to about 1 gram per minute, even when large amounts of carbohydrates are ingested. However, combinations of carbohydrates, coined multiple transportable carbohydrates, particularly glucose and fructose, that use different intestinal transporters for absorption, have been shown to result in higher oxidation rates. This seems to be the way to increase exogenous carbohydrate oxidation up to 1.7 grams per minute. Combinations of carbohydrates are recommended to increase glucose oxidation at a rate of more than 60 grams per hour. Carell and Druckendrup reported that ingestion of a drink containing multiple carbohydrates, glucose and fructose, as compared to either a glucose drink or water, improved performance by 8 and 19%, respectively, in time trial performance following a 2-hour bout of cycling. Power output was greater throughout the approximate 1-hour time trial with the glucose-fructose drink. Single sources of carbohydrate, such as glucose, appear to be adequate when less is needed. Thus, check food labels for ingredients to ensure that the chosen sports drink contains such combinations. So you can see here the villi and the intestines contain different receptors for the monosaccharides, which may increase carbohydrate absorption when multiple sources of carbohydrate are ingested. So if we look at the different types of carbohydrate, and fructose in particular, most people can tolerate small amounts of fructose. It is found naturally in fruits and is an ingredient in some sports drinks. However, consuming larger amounts may pose problems. Because fructose is absorbed slowly from the intestinal tract, it can create a significant osmotic effect in the intestines, leading to diarrhea or gastrointestinal distress in some individuals. Oftentimes it's termed the trots for runners. Research has indicated that a 6% solution of fructose, when compared to a similar solution of glucose and sucrose, caused significant gastrointestinal distress and an impairment in exercise performance. The athlete should be cautious in using fructose as the sole source of carbohydrate before or during exercise. Sports drinks containing fructose included in small concentration. Fructose is generally not the preferred carbohydrate for post-exercise muscle glycogen resynthesis. However, relative to muscle glycogen repletion, little is known about liver glycogen repletion. So although there are few data available on liver glycogen repletion, it is possible that post-exercise carbohydrate consumption that includes some fructose and galactose may enhance liver glycogen accumulation. Additionally, high fructose corn syrup, although rich in fructose, is treated as an added sugar. The health risks of added sugars are covered later in this chapter. If we look at the types of carbohydrate, again, we're looking at solid and liquid carbohydrates here. In a review, Cermak and Van Loon concluded that there is no influence of the form in which carbohydrates are ingested. So liquid, solid, semi-solid, slurry on exercise performance. A small number of studies have been conducted that support this viewpoint. Importantly, an athlete's preference of carbohydrate form could have a greater effect on performance than carbohydrate form. So the important factors to consider in carbohydrate choice have little to do with form and a great deal to do with preference, dose, gastric emptying, and fluid and electrolyte needs. With low glycemic index foods, although research and on GI and performance has been conducted for 20 years, the data are far from conclusive. Several reviews from experts in the topic are available, and so Donaldson and colleagues concluded in a review that there is still a lack of agreement on the benefits of consuming low versus high GI carbohydrates on exercise performance. In their review, they noted that only 5 of 13 relative investigations of pre-exercise carbohydrate consumption measured performance, and that of 5 that did, performance only improved in the low GI condition in 2 studies. In a similar analysis, O'Reilly and others evaluated 13 studies on pre-exercise high versus low GI carbohydrate intake. Only 2 demonstrated a beneficial effect of consuming a low GI carbohydrate prior to exercise. However, this effect is attenuated if carbohydrate is consumed during exercise, which is common in most sports. Additional research is needed to evaluate the effect of pre-exercise low GI carbohydrate consumption on endurance exercise performance. So there are some studies that show some beneficial effects with low GI. There's greater running time to exhaustion at 70% VO2 max with a low GI versus high GI diet, as well as lower carbohydrate oxidation and higher fat oxidation with a low GI meal versus a high GI meal. Now, if we look at carbohydrate with protein, some studies have been conducted to determine the impact on performance of consuming protein with carbohydrate during exercise. McCleve and others found time to exhaustion to be 15.2% longer for female competitive cyclists and triathletes when a 3% carbohydrate or 1.2% protein supplement was consumed every 20 minutes compared to a 6% carbohydrate supplement. Several studies have compared the effects of carbohydrate supplementation alone to carbohydrate protein supplementation on performance following recovery from previous exercise. Betts and others had subjects complete a 90-minute run at 70% of VO2 max, followed by a 4-hour recovery period, during which they consumed either a carbohydrate or carbohydrate protein mixture. Subjects then ran to exhaustion at 85% of VO2 max, but there was no difference between treatments. The use of chocolate milk has been reviewed by Roy and others as a more economical alternative to sports drinks during recovery from exercise. Benefits to this beverage include a ratio of carbohydrate to protein in the range recommended by the International Society of Sports Nutrition, and its naturally high concentrations of electrolytes. It also affords a greater feeling of fullness compared to water or carbohydrate beverages. Individuality is something that should be considered. Probably the most important recommendation for the athlete is to experiment with different types and amounts of carbohydrate during training before using them in competition. Just as it's important for you to know your optimal race pace for an endurance event, so too must you know well how you tolerate different amounts and concentrations of types of carbohydrates. And just as you train your muscles to learn their capacity, you may be able to train your digestive system to know its limits. During training, experiment with various types and concentrations of carbohydrate both before and during exercise. So what is the importance of carbohydrate replenishment after prolonged exercise? There are possible applications of this question. One is the athlete who may be involved in a prolonged exercise bout, have a rest period of 1 to 4 hours, and then must exercise again, such as athletes who train 2 or 3 times daily. Benefits may accrue to anaerobic endurance, aerobic endurance, and resistance trained individuals. The second application is the athlete who trains intensely every day and must have an adequate recovery in the 1-day rest interval. A third application is covered in the next section, and it talks about the technique of carbohydrate loading. After prolonged exercise, increased levels of GLUT4 receptors in the muscle cell membranes help move available blood glucose into the muscles for resynthesis to muscle glycogen. There are many studies that have shown that ingesting carbohydrate during the rest interval between 2 prolonged exercise bouts improves performance in the second bout. This finding is comparable to the beneficial effects of carbohydrate intake during prolonged exercise bouts. The carbohydrate can help restore blood glucose levels, but may also be used to resynthesize muscle glycogen. In cases such as this, where the rate of muscle glycogen synthesis is important, high glycemic index foods such as potatoes, bread, glucose or glucose polymers would be the preferred source of carbohydrate, for they apparently lead to a faster restoration of muscle glycogen than does a meal rich and low glycemic index foods. For repeated prolonged exercise tasks with about a 4-hour interval, a general recommendation is to consume 1 gram of carbohydrate per kilogram body weight immediately after the first event and again 2 hours prior to the second event. Additional carbohydrate may also be consumed immediately before and during the second event. If we look at carbohydrate and protein replenishment after prolonged exercise, carbohydrate has been combined with other nutrients, particularly protein, in an attempt to enhance muscle glycogen resynthesis. Protein and some amino acids such as arginine may stimulate the release of insulin, which, if added to the effects of carbohydrate-mediated insulin release, could increase the rate at which glucose is transported into the muscle cell. Here, the International Society of Sports Nutrition, they have created a position stand on nutrient timing. Ideally, you want a 3 to 4 ratio of carbohydrates to protein. 3 to 4 to 1 ratio of carbohydrate to protein. And that may help to increase endurance performance and maximally promote glycogen synthesis. And we'll talk more about protein after exercise in chapter 6. So will a high carbohydrate diet enhance my daily exercise training? Most scientists and sports nutritionists who study carbohydrate metabolism in athletes recommend a high carbohydrate diet for most athletes, particularly endurance athletes. Because success in athletic competition is contingent upon optimal training, and for the endurance athlete, optimal training may be contingent upon adequate nutrition, primarily the ingestion of sufficient carbohydrates every day. Not all athletes, including endurance athletes, need a high carbohydrate diet all the time. So as you may recall, aerobic exercise training improves the ability of the muscles to use fat as an energy source, so they may be somewhat less dependent on carbohydrate for a given training protocol. Thus, a moderate carbohydrate diet may be adequate for trained athletes. For example, obtaining 45% of daily energy needs from carbohydrate might be considered moderate, as it is at the lower end of the AMDR. On such a diet, an endurance athlete who consumes 3,000 calories per day during training would derive 1,350 calories from carbohydrate, which is about 340 grams of carbohydrate. For a 60 kilogram athlete, this is about 5.6 grams of carbohydrate per kilogram of body weight. Although this is slightly less than recommended for endurance athletes, research has shown that such amounts may be sufficient to maintain training on a daily basis. There are some limited data supporting the concept of enhanced training following a high carbohydrate diet. A number of field and laboratory studies with athletes have attempted to mimic actual sports conditions. For example, one group of soccer players improved performance on an intermittent exercise task designed to mimic physical activity in a game, while another group improved performance in a standardized intermittent running task and a run to exhaustion. In other studies, runners were able to endure longer on a treadmill run to exhaustion. Swimmers were better able to maintain 400-meter swim velocity, and triathletes experienced a significant improvement in treadmill endurance following 30 minutes of swimming, cycling, and running. Based on the available data, Edward Coyle, an international authority in carbohydrate metabolism during exercise, indicated that physical performance seems better maintained with a high versus moderate carbohydrate diet. In general, the normal carbohydrate intake of athletes in training studies was increased from approximately 40 to 45% to 55 to 70% of the daily calories for varying periods, but usually a week or more. This level of carbohydrate approximates the upper levels of the AMDR for carbohydrate. In summary, as Cole notes, athletes do not train hard every day, so they do not require a high intake of carbohydrate every day. Nevertheless, a diet rich in healthful carbohydrates not only may have several major health benefits, but also help guarantee optimal energy sources for daily exercise training. Moreover, Louise Burke points out experts in the field of energy metabolism indicate that there is no evidence that diets which are restricted in diet, such as the zone diet, discussed in Chapter 5 and 6, enhance training. Carbohydrate is the major fuel for most athletes in training. The slogan, "Train high and compete high," refers to the concept of training and competing with high carbohydrate intake. And these are just some dietary strategies for rapid muscle glycogen restoration. You want to consume carbohydrates immediately after and about every 30 minutes for 4 to 5 hours post-exercise. Consume about 0.6 to 0.8 grams per kilogram of body weight, and consume carbohydrates with a high glycemic index. You can consume it either as liquids or solids. Other dietary strategies for rapid muscle glycogen restoration: consume lean protein with your carbohydrates. So there should be a carbohydrate protein ratio of about 3 to 4 carbohydrates to 1 protein, or 3 to 4 grams of carbohydrate for each gram of protein. This full procedure is not necessary if there is ample time for recovery, but consuming a carbohydrate and protein combination immediately after exercise is recommended. We have reached the finish line for today. We will be continuing our discussion in Chapter 4 tomorrow. I look forward to talking with you soon. Thanks. Have a great day.